Yes? Just to understand a little bit the students, who has done fluorescence microscopy? 1, 2. Okay. So who has done cell biology? Okay, I got okay. What's that?
Isn't it the same question?
No, you could have been doing single molecule microscopy, in vitro reconstitutions, and never touch a cell, right? Could happen. Okay, because of that, I won't tell you anything about cell biology. I won't tell you anything about fluorescence microscopy, although my talk will be about cell biology and fluorescence microscopy. So it is true that I've done a lot about structural biology. I've integrated studying something called Clathrin-coated vesicles, is the way that cells eat. In fact, I've done a lot of that work. I started that work when I was a postdoctoral student in Steve Harrison's lab. But I won't talk really today about that, although we have isolated the proteins, got the genes. You heard a little about the reconstitution of viruses. We have done the same with the Clathrin coats. Some years ago, we did crystallography. We moved the structures. We were able to see this by cryo-EM. We did reconstructions. These are objects of the scale of the nuclear pore. So we're talking also of 1,000 angstroms. Not as many molecules as the nuclear pore, but we're still in the hundreds. But what I want to do today is do a little bit different. There has been a revolution in cell biology and in imaging. There's a new form of microscopy I would like to address today. It's a form of microscopy that you can push it to see single molecules if you want. You can do single molecules inside cells. You can see now dynamics in cells in real time. You can image the whole volume of a cell. You can even do it in a cell within a tissue. Now, this is a microscope that was developed by Eric Betzik and his colleagues at Janelia Farms a couple of years ago. It's called a Lattice light sheet microscope. And I was very fortunate to encounter him because I was participating in a meeting that I had been invited in structural illumination to participate. And I actually met him and we talked about the instrument, and eventually he said, well, I asked him, well, any chance that one can use your instrument? And he said, well, if you write a grant proposal, I'll think about it. So I wrote the fastest grant ever in my life. It took 2 hours. I sent it by email, and 30 minutes later, the phone rang. I said, well, you're in. You can come and do experiments. That was very cool. So that was transformative to us because we ended up going, did some experiments. I took people from my lab. We spent several weeks, full-time, completely immersed. And at the end, I said, well, I want this instrument, right? So I said, well, how do I get one? I said, well, here are the prints, and you can build it. Okay? And then I was ready to walk out. I said, actually, that's a terrible idea. You will never be able to build it. It's too complicated. So we'll help you. So that happened. And this is an instrument that has lots of optical components. A normal microscope has like 3 optical components. This has about 90. And you will see this in a minute. But it's very cool. It's really a nice instrument. And so what I'm going to do today is I want to walk you through first how it works. Second, give you a few examples in cell biology, how we're using it. And then I'm going to push the envelope to see how we're using this to now use analytical techniques to follow things in the cell. So I'm going to show you, you heard about viruses. So I'm going to show you that we can actually see viruses. We can see viruses entering cells. You just heard about VLPs. I'll show you that we can check VLPs going into cells. You heard about nuclear pores, so I just added a slide on nuclear pores so you can see that in a live cell. And then I'm going to show you how we can also follow cell biological properties. I'm going to show you one in particular. It has to do with how you regulate the size of a cell when the cell divides. I'm going to do that in a cell in a culture. And then I'm even show you it's possible to do this type of imaging in a tissue. So I'm going to do that in a zebrafish. Now why I'm going to do all this is because I need your help. We need your help. So this is so new. The problem is that we don't have people to help us do analysis, right? We're stuck. So we're too excited. Our files are gigantic. We have tons of data, but we don't know how to analyze. So I'll give you a glimpse of what we're doing, but we desperately need help, especially for the students here in the audience. So if you want to come and participate, you're most welcome. So let me start first by pointing something also in cell biology. You heard about introducing pieces of RNA or DNA into cells. That sometimes that's called transductions or transfections. That's a way to express things ectopically into cells. I now don't allow to do that in my lab. We've switched out of that, and at least for cell biology, we're now using something called gene editing. So now we introduce into the genome, into the cell, the modified protein that we wish to see. For example, if you want to put a fluorescent protein, we don't transfect that fluorescent, we introduce in the genome. You will see in a minute why that is important. We can now express at physiological levels what used to be vast amounts, and you didn't have control. That makes a big, big difference in analyzing cell biology. So this is going to be important. You will see that as I go through the talk, we are approaching now the possibility of doing biochemistry at the single molecule level inside a cell, which is also remarkable. So if you're interested in kaons, K-off, assemblies, dynamics, stochastic phenomena, it's a way to do it. We now have the technology to do that in vivo, in a cell. And then the technology I'm going to be talking about is this instrument, is the Lattice light sheet microscope. This is the invention that Eric just made recently. So when we do microscopy, the normal way we do that, we have an objective. We have light, is like my pointer, right? I have a beam of light that's going all the way to the wall, right? Usually it's a Gaussian beam, and you illuminate the whole volume of the cell. You can focus somewhere. So you can focus on a particular plane, that could be a confocal microscope, and that's where you image. Now, that works fine. It's a standard form of microscopy. The bad news is that you illuminate the whole cell all the time. So if I want to get a plane, and I illuminate one plane, let's say this plane here, I illuminate my cell, then I go to the next plane, and I have to illuminate the whole cell again, even though I'm imaging only one plane, and so forth. So if I'm going to do 100 planes to collect, let's say, every 0.5 microns in my Z steps, I'm maybe going to illuminate 70 times my cell. So if the cells have fluorophores, they bleach very fast, it's a disaster. Or if I'm going to be going through cell division, it's very bad because it's very toxic for the cells. There's another way to do things. I can go and illuminate only one plane, and that plane can be just at the surface of the glass, this red stuff here, right? So that's something called total internal reflection fluorescent microscopy. Huge name, TIRF. And the trick is that you come with a beam of light, and instead of going normal to the sample, now I'm going to come on an angle. And it's going to be a shallow angle in such a way that the light will be completely reflected. And I create an evanescence field very close to the glass. Usually that evanescence field is maybe 200, 250 nanometers. So that's what I illuminate. Nothing else gets illuminated. So now I get a very good signal here. I can see molecules, but I'm not illuminating the cell. Plus, I have to put the cell on glass, and that not necessarily is a good idea. This new method will now give me a plane of light through the cell. Let's imagine it's this dotted line that you saw in cross-section. And I'm going to be imaging in this direction. So I'm going to have an objective that creates this plane, and I have an objective that images on this plane. This plane, as you will see, is very narrow. It's about 400 nanometers in thickness. So it's equivalent close to the TIRF, so I can do single molecule. And the cool thing is that I'm not illuminating anything in the cell except my plane. So if I now need to collect the whole volume, I'm imaging one plane, image. The next plane, image, etc. Right? I can do this very fast, can be milliseconds per plane. I can collect the whole volume of the cell in maybe a second. So I'm fast enough that I can start to follow molecules, diffusion, and things like that. It's possible to do that. And at the same time, since I'm only illuminating the plane when I'm imaging, if I'm going to do my 70 planes, I only illuminated the volume of the cell once, right? For each plane was one plane, right? So now I gain maybe a factor of 70 or 100 in illumination. And it's very dramatic effect. So I'm going to show you a movie first using the standard confocal microscopy compared to one of these things. It's going to be a cell that is expressing a dye that levels the mitochondria, which is how you get energy. Mitochondria are all over the cell, and you will see a view. We're going to image in this direction. We're going to be imaging this way. We're going to be imaging from the top. I'm going to show you the projected views of the whole cell. That will be one movie you will see. And a similar cell expressing the same marker by using the second method, using this trick of the Lattice light sheet. So in the first method, with the first, actually before I do that, just to give you the physics of it. So in the normal imaging, you have the objective, and you come with your beam, you illuminate the back focal plane, you use the objective as a Fourier transformer, you get your conical beam, and that's where you image. This other way, what you are going to create is a pattern of light in the back focal plane, and that pattern of light, when it gets Fourier transferred by the objective, creates a Bessel function. It's a Bessel beam. Very complex, lots of flares. And then the trick is we're going to generate a number of these guys, one next to each other. And since I'm using coherent light with the laser, I can bring these beams close enough that all the flares will disappear by destructive interference. And the only thing I'm going to have at the end is the central part. And most of the energy, like 95% of the energy, is in the center. So I'm going to show you a view of this, and then I'll show you the data. So this is the cross-section of this beam. So each one of these guys here, maybe we can bring down a little bit the light. Is that possible? Sorry. Okay. So each one of these beams is now a cross-section of these Bessel functions. There are a lot of things here. You'll see now the beams coming inward, and you will see the destructive interference. And so you end up having just the central beam. At this point, that's about 90% of the energy, 95% energy. And then we have a mirror that allows us to sort of move very fast these 31 tracks that we have, and that's how we create the sheet. So that sheet ends up being about 300 nanometers in thickness, and we can make a length that can be several microns. In fact, I'll show you that we can go and image a tissue. In that case, it's about 80 microns. So we can go in this dimension, we can go maybe 80 microns. In the dimensions from you to the wall is another 80 microns, and we can go about 60 microns. So it's a whole volume. So we can do tissues, we can do many cells, and it's all this subcellular level. Okay, so this is then the objective that creates the sheet, and that's the objective where we image. Okay, this is the instrument. So lots of complex optics now. And the sample will be located in this region here, which I'm going to enhance. So this is the objective that illuminates, and the sample is in this location here, and this is the objective where we look. This is a chamber where we have there's a sample holder, like it would be like from an EM, and the sample goes here. This chamber, we call it the jacuzzi chamber, because the cell needs to be bathed and be in the proper conditions of pH, oxygenation, etc., right? Okay, so, and we put the sample there. Okay, so now I'm going to show you a comparison movie. So this is going to be the cell that is being seen from the top. This yellow stuff is the mitochondria. There are many, many mitochondria, and you're going to see a second cell. Same idea here. The central part that is dark, that's where the nucleus is located. That excludes mitochondria. And you will see the two movies running at the same time. This first movie here is generated with 77 planes, and the second movie, it has 160 planes. It's even twice the number of planes. And you will see in the left part here a trace where you will have here the signal to noise for the signal, for the fluorescence, and here you will have time. So just have a look and see what happens. So this is gone. This is still running. Okay, thanks. This is still running. So this signal was gone after maybe 20 of these scans, which is already considered a lot for cell biologists. This one, we went to 400 of these scans, and we stopped the movie because we simply got bored. Okay, so you can now do these very, very long images, and you can do, remember, this is collected in volume, right? So we're collecting the whole volume of the cell. And we went, in this case, a huge amount of cycles. Okay, so I'm going to show you first some examples just to give you a feel of the biology of what we can do, and then I'll go to more specific where we can do some of the analysis. So this is the first one. This is a cell, and we're putting a protein, which is called CD9. It has this thing, EGFP, so it's fluorescent, and this protein is being targeted to the surface of the cell. It was gene edited, and you can see that this cell has these spikes. These spikes are called filopodia. So we knew that cells have these projections. They have actin in the middle of these things to project, but we have seen in the field these things when you, for example, could have done an electron micrograph in one plane, you could see these projections, but it was not understood how many of these things existed, whether they were rigid or not, how they are moving. I don't have them here on their growth, but you can see that they're oscillating. We have no idea whether it's synchronized or not. These guys are sensing environment in the cells. They're very important. If you don't have these things correctly in cells, they don't invade correctly, and they don't migrate correctly when you're developing a tissue. Okay, so this is an example, right? In this particular movie, these were, I think, 15,000 images that were collected, and 150 planes per volume stack. And this movie, it's about 7 minutes in duration. Okay, we are interested in cell division, so we took fish and we put another protein that goes to the membrane of the cell. The name really doesn't matter, and I'll show you that later and how we use that fish to study cell division. But the postdoc that did the work in Berik's lab had never seen fish, got all excited, started to look around the fish, and found those two cells. So those cells are macrophages. These are the cells that eat, part of the immune system in the fish. Again, it's a volumetric imaging, and you can see now all the rufflings that are occurring in the cells, and I'm not exactly sure what they are doing. Maybe kissing, I don't know. But you see now biology in action, right? So there's no reconstitution here. This is really how the system works, and you can imagine now one can use situations like this to extract back in data and trying to understand in this case could be the interaction of the cell with the environment, trying to analyze the properties of, for example, of the cell surface, migration, the response of different parts of the cell. Remember, this is again a volumetric imaging, and this was done in tissue. This is not just a cell in glass. It's actually, yep.
You mark, are there other cells that you don't see?
Yeah, in this case, this was the gene that was expressing the fluorophore was put as a transgene into the fish. Not all the cells are labeled. Yeah? Yeah. Okay. So when you think about cell biology and you're thinking about ribosomes, the ribosomes, you heard about ribosomes, sometimes we make a protein, and sometimes the proteins are made directly in the cytosol. Sometimes we make proteins that are membrane bound. And they're membrane bound, they're made in the endoplasmic reticulum. After you make your proteins in the endoplasmic reticulum, which is this lattice of membranes and cisternae, the protein has to go to, for example, the cell surface or go to other places in the cell. And usually, I mean, most of the time, when the protein is made in the endoplasmic reticulum, has to go to another compartment called the Golgi apparatus, and that's where it gets glycosylated, and then it gets driven to various places. So we know about this. We can do biochemistry in the cells. We can do immunoprecipitations. We can map different compartments that the protein would have seen, but we have never been able to track this in real time to actually see the process. And now I can show you this. So in this case, we took a protein that is called transferrin receptor. It's a protein, it's a membrane-bound protein. The details don't really matter. It's the protein that allows you to take iron into your cells. And we coupled to this protein the GFP so we can see it. And we added one more domain to the protein that allows it to be artificially stuck in the ER. And it gets stuck because this domain interacts with another protein we put in the cell. There's normally cycling between the ER and the Golgi. So whenever the transferrin receptor is made, it binds to that protein that is cycling and gets stuck in the ER. It doesn't come out. But we can now add biotin, which is a small molecule, diffuses into the cell, and within seconds, it breaks the interaction between those two domains. And now the transferrin receptor that was in the ER gets released, and now you can do like what's called a pulse chase experiment. You can track the protein in the cell. But we're going to track it again doing this imaging volumetric. And I won't be able to show you all the details. I'm just trying to give you examples. But believe me that we see all this in volume. And what I want you to notice is at the beginning of the movie, this is dark, that's the nucleus. This is the reticulum that you see. We just added the biotin, and suddenly there are spots that appear. So those spots are the carriers that are taking small vesicles that are forming very fast. They are self-assembling. There's a coat called COP2. It's self-assembling very fast. It's pulling out the protein into these vesicles, which is happening now. And then they're migrating to this stuff here. That's the Golgi apparatus, which is this very complex structure of cisternae where you're getting this glycosylation and then it goes out. So we're at the stage now where we have the results, and now we're trying to track these guys and see whether we can track it in 3D, at what rate they happen, how many times they occur, where they're going, and which direction. And there are proteins, then there's the cytoskeleton that locks this and takes you in certain directions. We're trying to understand it. Okay. I work on Clathrin, and I won't really tell you too much about Clathrin, but one of my dreams was, can I see Clathrin in action making coated pits, coated vesicles in a cell within a tissue, and that's what you see here. So this is another situation, another example. This case is a zebrafish. We label Clathrin with fluorescence. We're looking at the tail, in this case, of the fish, and what you see here, these striations, each one of these structures is one of the muscle fibers in the tail of the fish. And where you see dark in this region, that's where the nucleus is. And you see here several planes on this movie again that we took dynamically. And you see it's very active. And these are all the Clathrin spots that are forming very fast. Good. That says that therefore we can go to that range. Okay. So we heard about viruses. We heard about virus penetrating cells. And it was mentioned about viruses that have membrane envelopes as a way to enter. I'm going to show you actually a virus that doesn't have a membrane envelope. It's the opposite. So it's a solid virus, like you heard. It's also making like has icosahedron. And this is Rotavirus. It gives you diarrhea. It's an RNA virus. And this is a project I've been doing together with Steve for some years. So we have done experiments in the past that were spinning this microscope. But I'm going to show you a movie where we can now see the particle and track it in real time. And that has allowed us now, on doing the analysis of that, understand how the virus enters into the cell and how it penetrates. So the problem is that when you have a cell and you have a virus, which is solid in protein, this virus needs to break this membrane, the topology of the membrane. Right? It's not trivial how you do that. If it would be an envelope membrane virus, you have a membrane around the virus, it will fuse with this membrane, you get fusion and the context gets released. So that topologically is relatively simple. Here, we have a particle that is solid, protein solid, and somehow needs to make it. So now we have learned that this particular virus, what it does, it has several proteins. When it sees high calcium, which is in your gut, it's tight. When the virus touches the surface of the cell, we'll do it this way. Yeah, it touches the surface of the cell. I'm going to show you that it gets engulfed by membrane, and it gets surrounded by membrane, and then the virus is sort of like breathing a little bit apparently, and so one of the subunits has a capacity to bind to membrane and starts to poke teeny holes. Now the cytosol of your mammalian cells has, let's say, 10 micromolar calcium. It's very low. 2 millimolar outside, 10 micromolar in the cytosol. This virus is very unstable at low calcium. So the notion is that when you're poking these holes, the volume that you have here is such that the number of ions of calcium is quite low. It's maybe 1,000 perhaps. And they start to leak out to the cytosol. And eventually you get this massive conformational change in the whole virus. Everything opens up, breaks now the membrane, and gets released. Now, how do we know that? Besides thinking about it, is because I'm going to show you now a movie where in Steve's lab they took the virus, they remove a layer of the virus, and you can put back the virus, so you can reconstitute a virus which is actually infectious. It's not a full reconstitution in this case, but you can recode it now with protein that have color. And therefore, I can now have the core of the virus in one color, the surrounding part of the virus with a second color. So when the virus is intact, it will appear as a dual core particle. But the moment it does this step, it switches color, because I release the core that goes into the cell, and so you can see a color switch. And the second thing you can do is that when the virus is attached to the surface, it's doing Brownian motion, so you can even track it. You can get the diffusion coefficient. The moment it does the penetration, it goes into 3D motion. And now it's Brownian in 3D, so the pattern changes completely. Yep?
Is this a double-shelled virus?
No. Well, yes, sorry, it's a double shell virus, yes, sorry.
That's why you can do it.
Yeah, in this case, yeah, yes. Yep. Okay, so we did this, okay? So this is a cell which we have I'll come back to the second later, but we have the Clathrin signal is giving where the cell is, and the particles are here. We're tracking with this microscopy. There are two colors. The green guys turn out to be the intact particles. The guys that are in purple already have penetrated into the cell. You can, just by eye, you can even notice that the particles, there are green particles that are moving fast, and there are also purple particles that are moving fast. The green particles that are moving fast are particles that we're also taking in. They're not releasing contents, but inside what's called endosomes, which are vesicular contents, vesicular structures that are moving very fast inside the cell, but they're not allowing the penetration. And then you have the other guys, which are the purple. These are the ones that penetrated, and they penetrated from the surface. So how we know that? Because we can track these guys. And you have it here, right? So in the bottom part is the whole movie. We're tracking out every single particle in the cell that is being attached, right? We have full mapping of this. So it's not just a region that we have selected. And then that's an enlargement. So let me just rerun it again. And then the central part, you have an enlargement of one of this particular one guy. And you can see here, it was doing the Brownian motion in 2D, and then goes away. So now you see infection in real time. Okay. So we heard about two things, VLPs, and we also heard about nuclear pores. So I couldn't resist the temptation to include this movie. So this is a movie where we have the nuclear pores. They're labeled by gene editing. So each one of those yellow spots is now a nuclear pore, this cell that we're tracking. You can see there's even some movement going on. So it's not really stuck in the nuclear membrane. And then we made VLPs. In this case, the VLPs that we make are for JCV. So these are 3 proteins. There's no nucleic acid here. This is a DNA particle, but it's also a small particle. And these guys, however, can bind and do enter the cells indistinguishable from the native particle, even though it's recombinant. And what is shown here are the trackings. We can track now the particles. These are the particles that have entered the cell. They were taken into the endocytic system, arrive to the ER, released from the ER, and the particles that you have here are particles that are at the surface of the nuclear membrane. And I don't know whether they are trying to enter into now the nucleus, what they are doing. But again, it's the first time you can see particles that are moving around in the surface. Okay, so far I'm giving you thousands of examples, just so I want you to get a feel that it's possible, what's possible to do with the microscope. But you see I'm a little qualitative because we're missing the level of quantification that I think we really should have here, and that's what we're trying to aim, right? Okay, so let me go now to endocytosis a little bit. So since we're doing history of science, I have to go to 1980. So in 1980, it was known that you have a cell, and you had at the surface of the cell there were these invaginations. The diameter of these guys tend to be about 1,000 angstroms, 1,200 angstroms. At some stages, they're still connected to the surface, and that's called a Clathrin-coated pit because there's an electron-dense material that's projecting to the cytosol. That's the Clathrin coat. I'll show you in a minute. And they trap things inside. So they trap, for example, the transferrin receptor I just mentioned, this membrane protein. They also trap other proteins, LDL receptors for the bad cholesterol. That's how you get cholesterol into cells. This section of microscopy has been labeled with an antibody that has gold particles of different size. So we had a section of EM, and we labeled just to know, this was a way to learn that these structures capture in a very efficient way the cargo. Now, these structures have several hundred proteins that are responsible for bending the membrane, curving, and then cutting. And the whole process takes maybe 45 seconds to form. So I've spent many years studying this and other people. I'm going to show you in a minute an animation that summarizes all this, so just to put it all in context. So we look at these objects that form in the cell surface. So this is driven by Clathrin, which is the polymerizing molecule that makes a lattice that looks like a cage, like a basket. And there's another protein that is important called AP2, which is the linker between Clathrin, which is membranes, is not membrane, it's a cytosolic protein. The adapter is also a cytosolic protein, but the adapter likes to bind to Clathrin, also binds to lipids, so that's a way to bring it to membrane. It also binds to the cytosolic tails of these cargos that I just described. So these guys self-assemble, they deform the membrane, and then there's another protein that comes called dynamin, which is what breaks the neck, the connection between the vesicle and the membrane, and then the whole thing falls apart with another reaction which is also energy dependent. So when you look at by EM, this coat has this appearance, right? So these are lattice of hexagons and pentagons. The pentagons is what gives you the curvature. And the more hexagons you have, the larger the object is, the less pentagons, the smaller. And so this is a partial assembly, and after you finish, you make a coated vesicle. So we have illustrious people here. We have Mike Levitt, we have Roger. So if I would take their brains and would make a pancake out of their brains, which would be, unfortunately, an irreversible thing to do, but if I would take their brains and make a pancake, the surface area of their brain would be about, like a soccer field, right? I think it's like 7,000 square meters. And in 1 hour, if they would be thinking and not falling asleep when I talk, okay, now he's awake. Now, so if they weren't thinking, they have synaptic vesicles that are coming all the time. You have lots of membrane recycling. So all the membrane is being internalized through this Clathrin machine. It's a very efficient system to bring in these thousands of square meters. That happens in all your cells in your body, plants, animals. As long as you have a nucleus, that happens all the time. And so this machine represents about 1% of the surface area of your standard cell. So it's a very efficient system to bring things in. So I'm going to come now to start to describe this in a little more detail in the context of what we're learning in the biology of these systems. So with Steve, some years ago, we solved the structure of this Clathrin coat. This was by cryo. This was about 10 years ago. That was done by Alex Foding. He was a biophysics student in the lab. And what you see in blue is the Clathrin part. In yellow is one of the subunits of the Clathrin. It's called the light chain of Clathrin. So this structure at that time was done at 8 angstroms. I think Alice collected about 9,000 particles, painful as hell in those days, by hand. You don't do that today by hand, right? Steve, do you remember how many hours of CPU was this? It was 3 weeks, right? Doesn't even remember anymore. So it was 3 weeks of CPU time to solve this structure, a little bit different than today. But this gave us the mindset, the place where we can do things. And we had some crystallographic data from bits and pieces of the system, so we mapped them. So the Clathrin molecule is a molecule of about 640 kilodaltons. The whole structure is 22 megadaltons. So that was it. And we had it at 8 angstroms, so we were able to trace the whole, almost the complete molecule of Clathrin at the time. But I won't go into that part today. It's just a framework, because I want to show you simply a quick animation that we have that summarizes where we are today in our understanding of the system. So, and then I will continue with the microscopy. So we have an event where the cytosolic components come to the membrane, and that's how we initiate. And then you start to polymerize the Clathrin molecule. It bends the membrane. It brings the adapters. You bring the cargo. There are a number of accessory molecules that allow you to stabilize the structure. If you don't have them, these things don't work. We can reconstitute these things in the test tube. We can bring these molecules and make them in the lab. In fact, that's how we did the cryo. And eventually they pinch, and then you use this HSC70 to uncoat. And this process takes about 45 seconds to go through the whole system, and maybe 100 proteins are involved. Okay? Okay. Let's go through this. Okay. So there's no sound. Okay. So what you see here is now a simulation that will go through the process of assembly. And this is the Clathrins. And for all of these things, we have data, either single molecule, in vitro, in vivo, structures, biochemistry, binding constants. And the idea is that you have these adapters, which are the green. The transcriptins are polymerizing. What you have here is the transferrin receptor. Many receptors behave the same way. They are diffusing. They get trapped. And then you get this assembly until finally you close down the vesicle. You make a neck where these molecules are coming in place. This molecule is dynamic, as the GTPase, which makes a polymer. And eventually will squeeze. You get the hemifusion of the tube. This is a tube that connects the plasma membrane with the vesicle, and you constrict enough that you can go through the energy barrier, you do the hemifusion, and then the sort of very catastrophic fusion, which allows you to relieve the vesicle. At that point, you have now a vesicle. This vesicle changes its chemical properties, actually changes its properties of lipid composition. So you can bring in this molecule here in orange, that's called oxalin, and then in yellow comes another protein that is called HSC70, which is the encoding enzyme for the system. So for all these things, we have cryostructures and different conformations and binding constants and mutations, etc. So it led us to reach the conclusion of how this works. So this is about what it takes. It's about 1 minute. So now what I'm going to do is take a few of these components, put label, and show you how we follow these guys and what we see, and particularly in the context of this new microscopy. Okay, so this was a movie.
It looks like a Dali painting,
Like a what?
like a Salvador Dali painting.
Yes. That's correct.
Based on microscopy.
Sorry. This? Yeah, okay. So everything I show you here is, for example, in this, we label the protein with fluorescence, and this is the adapter, this AP2, and this is a time series that we have in the bottom of the cell, and you can see that each one of those spots is appearing and disappearing. Each one of those is a coated pit. Now, we cannot see them on themselves because they are below the resolution limit of the microscope. The optical microscope, you cannot see things below the diffraction limit. These objects are 1,000 angstroms, so the diffraction limit is 300 nanometers. You cannot see that. But what we can see is the amount of light that appears in that spot. And we calibrate the microscope so we know if you have a certain amount of fluorescence, it's 1, 2, 3, 4, 5, a certain number of GFPs. I mentioned that now we gene edit because this allows you to replace all the endogenous protein with the fluorescent protein, and now you can do proper countings, right? It's just, you get absolute numbers. And you can do mixtures of different proteins, protein 1 plus protein 2, etc. So this tells me, okay, it takes 1 minute to form, but it's a stochastic process. I mean, sometimes it's 1 minute, sometimes it's 1.1, 90 seconds, 40 seconds, etc., right? I can tell you how many molecules arrive when they arrive, right? So that's part of the information that came into the movie I showed you before. Since we also had the information on cryo and the structure, I know where the components go, what contacts they're making, what binding constants, and the timing. So that animation I showed you was the integration of all that. Now, this is a movie obtained with spinning disk. It's only the bottom plane of the cell. This was done maybe 12 years ago by Marcelo Ehrlich, who is now at Tel Aviv. And each image here took about 800 milliseconds to obtain. Now we get it in a few milliseconds, just to put context. And we couldn't count at the time. Now we can count. Okay, so now this is 2 cells. We're imaging now the cells, but now volumetric. And each one of those is a coated pit. So can we now track these guys? And the answer is yes. And what you see here is for that cell, this is the instantaneous tracking for that cell, and this is the cumulative. So each one of those tracings is one of those color pits. And what is color-coded in this case is the distance from the bottom of the glass to wherever they are in the cell, right? So not only we get the macroscopic distance, but we can also see the fluctuations locally. And I won't be able to show you, I don't have enough time to show that, but I can show you, we now know when the guy forms and they start to bud, they're attached to the membrane, they're not moving. And suddenly they move a little bit, and that's when they cut. We have a few seconds before they uncoat, and we can now track them as they're moving, and so we can therefore figure out the position from which they're cutting from the membrane. That tends to be about 150 nanometers apart. So we cannot see the object, but we can see the center. That's very tight, getting the centroid in 3 dimensions. That allows you to break the diffraction limit, and that we're doing with about 40 nanometers, 50 nanometers of precision. So we're way, way beyond the resolution of the scope. Okay, I'll skip this one because that's a detail of the experiment. But this is a dream experiment for us. For years, we wanted to now track cargo. I mean, can we see the cargo going into cells? The cells respond with signals. You can put hormones, you can put EGF, growth factors, insulin, but it has been very, very hard to understand where the molecule goes that is signaling in what part of the cell. So we can now visualize this. And as a test, we use, again, in this particular movie, is the transferrin, which is our standard molecule to follow. And what we have here is a cell. So this is the outcome of a cell which is oscillating in volume. We're tracking 2 things right now. The Clathrin, which is in green, and then we have in this orangey thing, That's the transferrin. The transferrin we added to the cell is a transferrin that's fluorescently labeled, and each transferrin has one dye, one fluorophore, like I think it was a Cy3, 1. And the transferrin tends to bind as a binds as a monomer to the receptor dimerizes, so we can track the presence of the dimers. Right? So whenever we see these giggles, actually that's the transferrin that went into the cell, and we're actually tracking as it's binding and it's entering into the cell.
Is
Yeah.
it localized?
Yeah.
Is it a heuristic?
Is what?
Is the transferrin localized?
Yes.
Taken up in a localized
Yes.
spatially
Yeah.
involved
Exactly.
way?
So
Fine.
yeah. So now, okay. So that's a little bit hard to see. So let me now separate this analytically. So what we have done is we have code that we wrote that allows me to track every single event of endocytosis. And you can internalize either by being trapped in a coated vesicle or not. Sometimes you can internalize without that. We can tell the difference because we can map where things are now. And that's shown here. So this is an average. So here I have the number of molecules per endocytic event, and this is the frequency distribution. And so if you focus in the green guys, there's a peak here in this distribution, which is 1.5 or 2. So that's basically the dimer I was mentioning. So the majority of events are entering as dimers here, right? And there are a few events that have a few larger content that presumably codes that are a little bigger, and they cut off beyond 20 is very hard to get. And in fact, if you now get the molecular size of the transferrin receptor, the cytosolic part, the cytosolic, sorry, the luminal part, which is where the big chunk of the protein is, and that's using the structure that Steve got, that's the upper limit that you get of packing that you can put in the volume of the vesicle. So it actually matches quite nice. Right? Now we have done this with toxins, we have done it with other molecules, and not only you can get the numbers, but you can also map the regions of the cell that are doing this. And it has to do with, when you start to think of evolution, development, contact signaling, it sort of matters, right? So now we're doing the equivalent to what the biochemist was doing of the single molecule experiment. So instead of doing the ensemble, now we're doing the single molecule in the cell. Okay, and just to finish the Clathrin part now, you can see now we can track every single Clathrin coat, right? So it's being tracked. So this is in the surface of the cell in this case. Well, of course, we can do that inside the cell as well. But this, I just wanted to present it so you can get a feeling of that. Okay, in the last few minutes, I'm going to switch now to a topological problem. So I'm going to go away from single molecules for a minute. It's going to be more macroscopic. But just to bring you an example of a computational problem, which is if I have a cell, let's say I'm a cell, I'm in interface, I have shape. I'm like, it can be like an elephant in shape, right, or a person. When I go into mitosis, I'm going to become a sphere. And as soon as I divide, the sphere becomes into two daughter cells. So they go into cell division, and then I cut the connection between the two cells, and I start to grow again. So there's a topological question here, which is if I have the cell that's in interface and has shape, when I go through cell division, a process that takes about an hour and a half, do I maintain my volume or do I change it? And second, if whatever I do, change or not volume, what happened to my surface area? Do I maintain it or not? Now these things matter again because when you have, for example, stem cells, they divide asymmetrically. It matters how you put things in your daughter cells. It matters where you in a particular region in the cells. Now it has been very, very hard to study this because there were no methods to image, right? I mean, the imaging methods I described before this new microscope was just like a one plane, 2 planes, 3 planes, or it was so toxic that you would kill the cells and you couldn't track them when they were dividing. So I'm going to show you this new technology. We can follow now the volume of the cell, the surface area, the activity of membrane traffic, and the challenges that we have, of course, to do the numbers out of this. Okay, so this is how a cell divides. This is the standard textbook image. You have a cell divides. In green is the chromatin that is labeled here with a protein called H2B. It's a nucleosome that Roger will remember. He figured out how the nucleosomes were organized. So this is H2B labeled with the GFP. And this form of microscopy has a little fluorescence and also phase microscopy. And we're just looking at projection. Okay? So this got accelerated a lot, of course. But the process I'm going to show you is just before the cells divide and when they divide. So that's about an hour and a half. Okay, so the question then, you have my cell and interface, and if they would have, let's say, equal volumes, do I maintain my membrane? Do I fold it? Do I take it into the cell? Do I spit it out of the cell? What do we do? So the way we address this is by putting a marker that goes into the cytosol, and then we wrote code that finds the boundary between the cell and the outside of the cell. It sounds easy to say in words. It's a pain to do by calculation, because you have a very noisy signal, and you have to get the gradient of the intensity vector in 3D to find the boundary. Particularly hard when you have two cells in contact, like you have it in tissue, but it works. I'll show you that. And then for the membrane, we have another protein that you can also go directly to the membrane, gives you a very acute boundary. And so in that case, you can take the second derivative, for example, and then you get the sharp the sort of the center of the membrane gets very sharply determined. So even though you are below resolution, it's very tight. You get that number. Okay, so what you see here now is views of cells that are dividing. So the green guys is the Clathrin activity. These are the spots. Now we can see them that they stay as the cell divides. And you can see as they divide, well, there are even funny blebs that appear. Okay, I'll tell you in a second what that is. So this is another way of seeing these cells. So this here you can see the volume. Here you see the spots. And you see this blebbing. This blebbing was discovered by this form of microscopy. It was thought that this blebbing was cells that were dying, because in culture you see blebs, cells bleb. But when you follow them now this way, it turns out that the cells that bleb tend to be the cells that are doing this last step in cell division. It's called telophase. And so every cell that does telophase is blebbing. And the blebbing is because it turns out that these spots represents endocytosis. The cells, when they go into the volume, they shrink. And as soon as they go and they divide, all the vesicles that were inside trapped are appearing back into the cell surface so fast that the cell doesn't know yet what to do with its membrane. And so this extra membrane are these blebs that you see here. Okay? So it's not that they're dying in this particular case, it's just membranes that just came out. Okay, so this is another way of seeing the cells. So now you see just in cross-section, and you can now see the blebs. I think it's easy to see that. And in this representation here, now you can see the blebs in the volumetric imaging. It's, I think, quite dramatic. Right? Remember, these are cells, and we're imaging the whole volume of the cell as the cell divides. Right? Yeah. Okay, so now I'm going to show you in a cell in the right. You will see a time series, and then you will see that as the time series goes, there are three parameters that we're following. We're following the volume, the surface area, and pits means how many pits we're making per unit time. So let's have a look, and you will see that the cell starts to divide. We have a certain amount of, for example, volume, and the volume will be more or less constant. It's quite expanded here, but it's essentially constant through the cell division. The surface area actually goes up. It was low and then jumps. And there's a quite dramatic jump. The jump happens when the cell divides. That matches to the blebbings. And this portion here, the details don't matter, but effectively what it means is that the endocytic activity is constant through the cell division. There was a matter the details don't matter, just you're getting cell division. Okay, and I'm going to finish with this. This is, I think, the most difficult experiment we did, which was, okay, let's take a fish. Let's make it transgene for the protein that labels the plasma membrane. Remember the macrophages that were having fun? Okay, same fish. But now we're going to look at the eye. So we waited for 18 hours after the egg was fertilized. Then we put it in the microscope. So we already have a small embryo. The eyes are still not well developed. These are just the beginning of making the eye. And there are hundreds of cells here, right? So you cannot tell anything from this except there's a blur of stuff, right? So in this movie, just to put context, is about 300 gigabytes worth of data collected in 90 minutes. So that's 100 DVDs about. And so we have to now get information out of this, right? So the first thing, of course, you have to have decent quality data. So I'm going to show you that this blur that you see here is pretty good, actually. I'm going to show you one plane in this movie so you can see it. This is about 90,000 images that were collected, right? So this is now the plane. So this is now, each one of this here is the outline of the cell. Sometimes if you look carefully, you can see two outlines because the cells are not in complete contact. I mean, I always thought that membranes were in total contact. Turns out that they are separated. They're not attaching completely. So Sometimes you can see double lines. And so then we wrote code that can track now both the volume and the surface area in real time for every single cell in this system, right? On doing that, then we can identify by doing this now cells that are dividing, and it turns out that in this particular movie there are 2 cells that are dividing, which are these 2, and we track this automatically. This is all done by hand. So Goku, who developed the code, at the beginning we didn't have the code and we did it by hand. So it took about 12 hours of brain CPU time from Goku. So he took 12 hours to do tracings on the cells that he found were dividing, so then we can track it, right? Now it takes about 15 minutes and we can do automatically everything in the system. Right? Okay. And turns out that for this particular situation we have volume that is constant in cell division, and like in the cells that we had in isolation, there's also this jump. So this is the mitotic cell, and then it goes up, and in going up, when the cells are starting to bleb, that's when the membranes are coming out. So I'm going to jump this. So so far, all the examples I gave you, I am trying to convey you where is the cell biology today, at least a particular form of cell biology, a particular form of imaging. We're desperate for people that can help now bringing in the analytical part, because it's a bit hard to, as you can see, handling this on a qualitative basis. The last example I'm going to give you on imaging, and I'm going to end with that, is we can use the same microscope, but now we can push the limits of resolution. We can also do structural illumination, and so that brings the resolution even higher. So I'm going to show you an experiment that was done by Dong Li, that was in Eric's lab, where we put again our Clathrin system, and you will now see that instead of being the Clathrin spot as a spot, actually will appear as a ring. We have 1,000 angstrom in diameter particle, roughly speaking. The resolution went down to maybe 10 nanometers, right? So we can now see details, and you will see that. And for the people that are doing biology here, we were interested in knowing whether the actin was in some way connected to know or not to the formation of the coated pits. So we have a marker for actin, and you will see the quality of the data, and we did as well a little bit of analysis here. Okay, so that's how the movie looks like. So red is actin, so it's a marker for organized actin, and the green is our Clathrin coats. And so I'm going to enlarge this so you can see this a little bit better. And here you can see in this way of presenting it, just switching to black and white, you can see this donuts, and that's the outline of the coated pits as they are forming. And we learned from this experiment, there was a discussion in the field whether actin was always or not related to the formation of coated pits. And now we know that sometimes it does, sometimes it doesn't. So we were able to resolve those guys that were forming and had the actin signal from those that didn't, right? So what is shown here is the lifetime of our coats. I told you it was in the 40, 50, 60 time range. So they happen to follow an exponential decay. And then we can separate the guys by those that are connected with actin and those that don't have the actin, right? And so it turns out the taus are a little bit different, and it seems that the guys that have the actin finish a little bit faster than those that don't. So if you're interested, we can discuss a little more the details of that. But I was just trying to indicate with this that the power that we have now to do the biology. So today I covered a little of the biology I know most, which is the Clathrin code activity. I'll show you a few examples. I gave you a few examples also on virus. One case was virus entry. The other one was the VLPs, that we can see them already penetrated all the way attached to the nucleus. Okay, I pointed out that we can see nucleosomes the same way we can see other structures inside the cell, a number of situations that we can now follow. And then I gave you a practical example on how we use this microscopy for imaging and getting data on volume and surface area. I think this is the way to go, at least for cell biologists. It eats any other form of microscopy I know. I think it's much, much better than step microscopy by far. This is really fairly soft for the biology. And you can go quite deep into the sample. So I think it's going to be transformative. So there was lots of people that participated here. The key person in this work I present to you is Gokul. Gokul is a postdoc in my lab who helped set up the microscope and took over many of the experiments I described to you using the light sheet. And he has been working with some other people, in particular with Francois Aguet, who is a person who got a PhD on image processing. And so he set up the software when he was in Gauden's Danusa lab to do the tracking in 2D and then finish the 3D tracking interacting with us. And then there are a few other people that are listed here, in particular Kishore Mosanlanti, who got involved in helping us setting up the fish experiments I show you. And the people that are here, these three people are fellows in Eric's lab, Pichang Chen and Wesley Ligand and Tsung Lee. They all help us at the beginning when we started to do the instrumentation. I mentioned also the virus experiment with Steve. Currently that's done together with Eric Salgado, is a postdoc in his lab, and Gokul. The gene editing has been done by another fellow in my lab, Raphael Godin, who is now taking a job in France. And with this, I want to thank you so much for your attention. Please, can I turn on the lights? Yeah. Yeah, please.
I think in some of your examples, you show that this method can be used to track the movement of multiple molecules with different colors. So my question is, in addition to the locations of this molecule evolve according to time, is it possible also to learn some more detailed information about this molecule being tracked? For instance, whether this molecule undergoes some significant conformation change, or whether it reacts with other molecules along this period.
Yeah. So the easy one for me to answer is the interactions, because we can have, let's say, two proteins, two different colors, and I can do K on K offs, right? Or co-localization. That's not a problem. That's very easy to do. We are trying to figure out how we're going to do Fred here. I think we can do it, but we're just trying to set up the optics to do that. You and yeah. Let's make it.
Yeah. Fine.
Yeah.
So if you want to make some kind of medical imaging device here,
Yes.
What would, in terms of optics, what would be the main obstacle?
Medical means.
Yeah. So use it like in vivo in general.
So okay. So we are using this in vivo right now. We can do it in vivo, but the practical constraint that we have is that we have two objectives. And we need to put the sample between the two objectives. The objectives that we use right now have a working distance of about 2 millimeters, 2.5 millimeters. So you can probably put, I don't know, a hair from your nose, right? No. So the main problem optically, for the perspective of the microscope right now, is that the volume that I can put presented to the instrument is about 2 by 2 by 2 millimeter volume. Oh, well, okay, but you don't have to do it with fluorophoresis. I mean, you don't have to do it with gene editing. I mean, for example, you can put a labeled protein that you're following the uptake or something like that, right? Yeah, that you could do. So right now, I know I can do the experiment in fish. I show you. Next week we're doing our first experiment in C. elegans. It's another thing we can put there. I refuse to put a mouse right now in the microscope, in the instrument. The instrument is not ready for that. At some point it will be, but not ready. Yeah. Please.
Yes, I'd like to understand better your explanation of the origin of blebbing in the dividing cell.
Okay. Yeah. Okay. So a cell normally has almost homeostasis of endocytosis and recycling. So you take, let's say, the whole surface area of the cell is taken in per hour, and everything goes to the endosomes, and then you recycle back to the surface, right? You're in total control, and in a mammalian cell, that's a process that doesn't use calcium to control. It's not like in the neurons that are using calcium in synaptic vesicles. When the cell divides, when it enters into metaphase, it starts to round up, it shuts down the recycling, but it maintains the endocytosis. That was sort of what we discovered.
Build up more membrane.
You're building now more membrane inside. When you go to anaphase, you release this block, and that you activate a pathway. We don't understand it very well, but it's now calcium dependent. It's exactly like if you were doing synaptic transmission. You even use similar proteins, and now you have this abrupt fusion to the surface and happens. So I think that part of the bleb is in part because the cell is under high tension, right? You're putting lots of membrane, and it doesn't know yet how to dissipate this extra membrane, and that's, I think, the bleb. So that's the interpretation we have. Right? Yeah, I'm sorry, there was a question in the back, and then you. Yeah. Talk loud. Talk loud. I can hear. We can all hear you. I'll repeat the question.
First of all, thank you. It was beautiful. And
Thank you.
my question is,
Ask her to say that louder.
Well, thank you so much. Well, you have to understand, I always take the glory, right? But I don't do any experiments. So the fellows in the lab. Yeah.
okay, so my question is, how did you connect the scene to your light shape microscope?
Yeah.
And the second one is, how did it affect the speed of
Yeah.
the
Okay. Yeah. Okay. So the question I was asked is a technical question. How do I do structural illumination, right, in this microscope? Because what I show you basically was just a sheet of light that is moving. Well, the same way, I remember at the beginning I said that there were several pipes of light and we can move it. The way we did that is because we went to the back aperture of the objective and we have a device that projects the pattern into that back aperture. Well, I can put any pattern I want, including changing the pattern of the pattern, right? So if I change the pattern of the pattern, I'm actually now doing structural illumination, right? Which means that I have to take several images very fast on an object that has not moved yet so I can have the resolution, right? So right now, this instrument allows me to take an image every, the exposure is about 20 milliseconds. So I can take 5 images in 100 milliseconds. So if I put it in the biology context, it depends the problem you're looking, right? I cannot look at single molecules. They will diffuse too fast. But if I look at, I want to see a nucleosome, right? The nucleosome is there. It's locked. That's not a problem, right? Even I show you there was a little bit of drift in the cell. In the scale of the 1 second, that's not a problem. So you have to then set it for the right experiment. But it works beautifully. Yeah. Yes?
So you started your talk with the statement that, of course, if you illuminate the cell too much, you cause a problem. And therefore, this is how you justify the light sheet. But in the end, you went to structural illumination, which includes much
No,
more.
sheep. Well, okay. Yes.
No, this is what you're now
Yeah,
asking.
yes.
You, of course, measure with light sheet the Clathrin movement. And you could have done the same with much more light, let's call it, with super resolution microscopy.
No. It's like because the problem is that if I want to do super resolution, let's say structural illumination, it now depends what is the question I'm trying to answer, right? If what I want to do is the organization of a structure, right? I can afford, and it's not moving, I can afford doing n images, right? And I can arrange my imaging such that I won't bleach in the period of the experiment, right? I can do that. I can do that with nucleosomes, it's fine. Even though the copy number in the nucleosome is very low, let's say I only have on symmetry of 8, whatever number of proteins you're looking, right? It's still okay. But if the guys will be moving, it's not okay. Or if I will have less copy number of that, it's also not okay, right? But if I want to get now the shape of a mitochondria against, let's say, how it touches an endosome, which are now bananas, right? They're very large. Now I'm okay, because I'm looking at the interface between those two structures, and it doesn't matter, right?
Fine, but the question I have is whether you have found different dynamics when you shed more light or less light on the cell.
Oh, yes. Oh, absolutely.
With Clathrin.
Also, so for example, in the Clathrin, Clathrin I'm not sure yet, because I don't know, but I can tell you with virus, right? So all this started historically in the lab because we were doing experiments with Steve following the Rotavirus, right? And we had the experience that we were doing the experiment and suddenly everything was frozen. The particles were not releasing. I don't have a good explanation yet for what's going on, but I think there's a photo reaction going on and somehow things are getting locked. And it was affecting properties in the host of the cell, right? So somehow the chemistry was transferred to the host. So, yeah.
Does it imply that
a lot of the research that was done with fluorescence that was heavily, a lot of light was shined.
No, was not.
Process
Was not.
not
I'm
the
arguing the
process,
opposite.
but
Yeah.
in other studies.
In the past, yeah.
In the past. Does it mean that we
Yes.
have to be very careful with the conclusions?
Well,
Fluorescence might
in my lab, they hate me right now for two reasons. First, because we're having this gene editing business, right? So we're repeating experiments that we and other people have done extensively. And it turns out that when you switch to these new conditions, both of illumination and of, in this case, the replacement, often we're finding big differences in biology, right? So I'm not, please don't go home and say I have to do everything again, right? It's not like that. But there's lots of biology. I think one needs to now be careful and reset and rethink and recheck and re-image. Because if you just calculate the amount of light, physically the amount of photons that you are putting in the microwave is brutal. If you're doing a STED who has done STED microscopy here? Good, I can now say anything I want about STED. So if you do STED microscopy, the brilliancy of the microscopy to do that super resolution is about 10 to the 5th the amount of light on the surface of the sun. That's what you're illuminating your cell, okay? No way you can have decent biology that way, right? This one is, I think it's still 4 orders of magnitude less than that, right? So it's very little, but still it's still a lot. But we are trying out, so we try to do experiments where, for example, I go and I say, okay, if I take a GFP, I'm going to put it in the microscope as a single molecule, how many photons I need to bleach it, right? So I use conditions where I know that if I would do continuous exposure, it takes me, let's say it takes me 5 seconds to bleach one molecule. Okay, that's constant exposure. Okay, so now that means that if I expose for 10 milliseconds, right, the probability I'm going to bleach goes way down, right? So that's how we organize the experiment.
Take further questions offline.